Hello, everyone. Welcome to today's live broadcast, Evidence Required by the FDA for Establishing Clinical Validity. I am Brenda Kelly-Kim of LabRoots, and I'll be your moderator for today's event. Today's webcast is presented by labroots.com, the leading social media site for science professionals. For more information, please visit www.labroots.com. Before we start, there are a few instructions. We want to hear from you, so please ask questions or leave us a comment. Answers are welcome too. You can do this by hitting the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We'll try to get to as many of your questions as we can, and we'll follow up with you if we don't get to yours today. Want a better look? You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, please let us know by clicking on the support button on the top right or use the Q&A button. We'll make sure we try to resolve any issues. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process for obtaining your credits. Now let's get right to today's presenter. We are proud to welcome Dr. Alberto Gutierrez. Alberto Gutierrez is the Director of the Office of In Vitro Diagnostic Device Evaluation and Safety at the Food and Drug Administration. Gutierrez has a long-standing career with the FDA, joining in 1992 as a reviewer in the FDA's Center for Biologics Evaluation and Research, working on vaccine adjuvants and method development for determination of the purity and structure of vaccine components. In 2000, he joined the FDA Center for Devices and Radiological Health as a scientific reviewer, becoming a team leader for toxicology in 2003, director of the Division of Chemistry and Toxicology Devices in 2005, Deputy Director of the Office of In Vitro Diagnostic Device Evaluation and Safety in 2007, and Office Director in 2009. He holds a bachelor's degree from Haverford College and master and doctorate degrees in chemistry from Princeton University. I'd now like to turn it over to Dr. Gutierrez for his presentation.
Hello, everyone, and thank you. Today I'm going to be talking about the evidence that is required for clinical validity at the FDA. So if you look at in vitro diagnostics, have a long history of being a major part of clinical decision making. It is believed that about 70% of all decisions are made with information from a in vitro diagnostic test. In the last 50 years, there really has been a revolution in the scientific methods and the tests that are performed, and things have changed tremendously. And molecular mechanisms of many diseases have been discovered, which allow us to better understand how tests or specific test pathways are connected to clinical diagnosis. But not all has been good news for in vitro diagnostics. It is still true that the biology and the clinical science are quite complex, nuanced, and not always intuitive. So it is clear that for many cases, the tie between what is being analyzed or what is being detected and what the clinical condition is still somewhat unknown, or how the tests are best used are unknown. There are lack of material and method standards, which means that there's still much to an art as to how one validates a test analytically and clinically, and that is going to be somewhat the discussion in this talk. And there is inadequate mechanisms for data sharing. So even though a lot of the information sharing is done through journal articles, really we still have a lot of the validation and what tests are still somewhat not shared and proprietary information. And the regulatory environment is actually not well defined in many cases, even though the FDA, as you'll see, has been regulating in vitro diagnostic tests. There are many tests that are done typically in the clinics or in laboratories that are not regulated by the FDA and that are not either well publicized or the basis for those tests are not well understood. So in terms of regulation, the FDA actually has been regulating tests as medical devices since 1977. Prior to that, actually, the FDA regulated some tests that were critical for drug, that is, things like the antimicrobial tests were regulated actually as drugs. And in 1976, the Congress passed amendments to medical device amendments, called the so-called medical device amendment, in which put all the regulation of in vitro diagnostics under devices. It provided a definition of what a medical device is, and as you will see, that definition clearly includes in vitro diagnostic tests. It defined standards to be used to assess what the validity of a test was, and we'll go through that with that standard carefully. And it provided a regulatory paradigm that was risk-based by which the agency regulates. Now, the regulatory, as you will see, the regulatory paradigm that was provided is not one that is a great fit for in vitro diagnostics, but it was flexible enough that we've been able to develop the mechanisms by which we regulate in vitro diagnostic tests in a way that we think is logical. And I will go through some of that. So the definition of what is a medical device, as you can see, is actually a definition that is very broad. It essentially means an instrument, an apparatus, an implement, machine, contrivance, implant, in vitro reagent, or other similar related articles, including any component, part of, accessory, which is intended for use in the diagnosis of disease or other conditions, or in the cure, mitigation, treatment, or prevention of disease in man or other animals. And so you can see this definition clearly includes in vitro diagnostics, either by clear reference to in vitro reagents or other similar parts of an in vitro diagnostic test. And so the definition puts all in vitro diagnostics, it doesn't matter where they're made, under the purview of the FDA. It provided a standard. We'll go through this standard more carefully later on. It suffices to know that it provided that there should be reasonable assurance that the probable benefits outweigh the probable risks, and effectiveness, that's for safety, and effectiveness that there is reasonable assurance that the use of the device will provide clinically significant results. And this is the basis of this standard, even under the different programs that we're going to look. It always refers back to this standard. The process by which, or the program under which we regulate is again, as I said, a risk-based. It defines 3 different classes of risk, that is risk to the patient. A class 1 or low-risk devices, class 2 could be more complex or higher risk devices, and class 3, which are the highest risk devices. And the paradigm defined that for all devices, there were some general controls that all manufacturers had to have, and we'll go through what those general controls will be in the next slide. That for the higher class devices, that is the general controls were the only things they had to have. They actually did not have to come to the agency for review before they went to the market. For the higher risk devices, that is class 2 or class 3 devices, it defined that there would be a pre-market review for those tests. And that pre-market review consisted in a different way, differently for class 2 and class 3. For class 2, the companies would have to give the FDA a pre-market notification, so-called 510K. And in that pre-market notification, the company would have to show the FDA that their device is substantially equivalent to something that is already in the market. And then for the most complex and high-risk devices, the applicant would, the manufacturer or developer would have to come into the agency with a pre-market application, and in that pre-market application, it would have to have data to show that the test was safe and effective. The Class 3 devices, in some ways, is closer to what people actually think the FDA does when it does things like drug approvals. That is, we do an independent review of that device before it goes on the market. There are major differences between the 510K paradigm and the PMA paradigm, and I'll be going through them as we go into talk. The one thing to note from the program as it was set up is that the Class 2 devices it really imply that you're comparing to your device to something that was already in the market. That means that for any new test that is coming to the market that is not equivalent to something that is already in the market, it automatically puts it into a Class 3. Now, currently we have ways to review those tests and to down-classify them, put them in the appropriate risk class as we review them. So there is a way for us to what we call a de novo classification. There is a way for us to deal with new tests and to end up in what would be the appropriate risk classification for that test. So what are general controls? General controls are things that we expect most manufacturers to be able to do. That is, first they would have to register a list with the FDA so that the FDA would know that they are manufacturing or they're making and distributing medical devices. They would have to follow good manufacturing practices. They have to report device failures to the FDA, and they have to have a system for remedying the device failures. That is, they have to be able to fix those failures that they find. Importantly for in vitro diagnostics, they have to have labeling requirements that are quite strict. Our labeling requirements come from drug requirements in some ways because as I said before previously, in vitro diagnostic tests were originally regulated as drugs. Now, for the Class 2 and Class 3 devices, we can have other controls that are not just the general controls that are expected for all devices. And those controls could be things like guidances and guidelines. It is importantly for most in vitro diagnostics, for most medical devices, there is a design control. And design control means that you design your test before you make it or you develop it, and you design what is it going to be for, what are the required inputs and outputs, and what do you expect out of it. And design control is quite important in device regulation. You have to have performance data. That is, after you design and you make your test, you have to validate that the test does perform as you think, and you have enough data to demonstrate how the test is going to perform. You have to track. You have to have tracking requirements. That is, that you make sure that you understand how the test is being used, and if the test is not performing appropriately, that you know that you're getting false results. You have to label the requirements that I spoke previously, and you have to survey, make sure that you understand how the test is performing in the post-market area. Now, pre-market reviews, as I said, we have two, depending on whether it's a Class 2 device or a Class 3 device. We do see different packages. For 510K, you essentially, we see them for new version of old devices, and for PMA and de novo high risk, we see a show of that the device is safe and effective. What are the elements of review? The elements of review for 510Ks and PMAs are, at least in general, similar. That is, we look at a test whether it is analytically valid. That is, does it it correctly detect the analyte or what it thinks the measurement, if you like, that is believed to detect, and does it detect it correctly? And clinical validity, that is, does it correctly identify the disease condition? What is the link between the analyte that you are detecting and the disease condition? And in general, and very important for all these, everything that we review is driven by the intended use of the test. The intended use of the test is crucial for determining what the test is, essentially, what its analytical validity should be and what its clinical validity should be. So analytical performance, this is really fairly well established. There are several ways to do it. It's well established in the sense of theoretically what we should do. It is actually very dependent on the type of analyte, the type of test, and what are believed to be the important factors that have to be controlled. But typically, people look at accuracy. That is, how well does the value relate to truth? Is there any bias? Precision. That is, how repeatable that answer is. If you can you do it again and again, and how much imprecision is there? Is the test specific? Are there other analytes, other things that may interfere with the accuracy of the test? And if they are, then typically one challenges the test under the different conditions that may produce incorrect results to understand what those possible interactions are. And depending on the test, lots of times the limit of detection are important. In other cases, it's not as important. But exactly what are the important factors that one looks at depends in many cases on the test, depends on the technology, depends on all the factors. And clinical performance, as I said before, is the yardstick of truth. That is, can the signal be turned into a clinical action? And when we look at clinical performance, then we look at clinical sensitivity and clinical specificity. That is, how often are you producing false results, either false positive results or false negative results. And for some tests, particularly those in which the prevalence of the disease is low, it's very important to look at what are the predictive values. That is, if you produce false results, how often are those? If you produce false results, then when you get a positive result, how often are those results false? And the same for the negative side. So in general, FDA review is just based on good science. As I said, the biology is complex. Many cases, the type of studies that one is doing to perform in either analytical validity or clinical validity are studies that are the best study one can design, not necessarily the perfect study. And understanding what are the limits of those studies is important in this area where you actually are making what you think would be the best study. It is very easy to bias the study in such a way so the performance looks much better than it actually is. So let me go back and just go through carefully on the safety and effectiveness at the bar that was set by the law and what does that mean. So safety, as is written in our regulation, is the reasonable assurance that the device is safe when it can be determined based upon valid scientific evidence that the probable benefits to the health from use of the device for its intended uses and conditions of use, when accompanied by adequate directions and warnings against unsafe use, outweigh the probable risks. The valid scientific evidence used to determine the safety of a device shall adequately demonstrate the absence of unreasonable risk of illness or injury associated with the use of the device for its intended use and condition. Similarly, effectiveness is the reasonable assurance that a device is effective when it can be determined based upon valid scientific evidence that in a significant portion of the target population, the use of the device for its intended use and conditions of use, when accompanied by adequate directions for use and warnings against unsafe use, will provide clinical significant results. Now, for those two definitions, one clear question is, what is a valid scientific evidence? And valid scientific evidence is also defined in the regulations. Valid scientific evidence is evidence from well-controlled investigations, partially controlled studies, studies and objective trials without matched controls, well-documented case histories and conducted by qualified experts, and reports of significant human experience with a marketed device for which it can be fairly and responsibly concluded by qualified experts that there is reasonable assurance of safety and effectiveness of a device under its conditions of use. What is clear from this definition, though, is that what we consider valid scientific evidence is much broader than what people usually think that the agency requires. Usually people think that the agency requires randomized clinical trials. And as you can see, we actually do not require just randomized clinical trials. And the type of evidence necessary or that can be used is quite broad. So I guess more importantly is the definition of what is not valid scientific evidence, because that kind of shows you the limit of where the agency, the type of data the agency is willing to consider. And we don't believe to be valid scientific evidence things like isolated case reports, random experience, reports lacking sufficient details to permit scientific evaluation, or unsubstantiated opinions. Now, based on the safety and effectiveness, you might be saying, well, how does safety and effectiveness relate to in vitro diagnostics? It's hard to see safety being an issue with a test that is done typically on samples that are not necessarily invasive, and how does that affect the safety of a patient? Well, we consider both the safety and effectiveness of diagnostics, which are tied in some ways, inseparable for diagnostics. And really what the safety and effectiveness is what happens to the patient, the risk to the patient with unrecognized erroneous results. That is, if a test provides a false, either positive or negative, what is the consequence of that false result to the patient? That's what we consider to be the safety and effectiveness as we look at in vitro diagnostics. So what does the FDA do? And the FDA bases its decision on the intended use of the device, and we looking at the performance of the device, that is, how many false results it provides, and what are the instructions. The FDA then weighs the benefits against the risk and determines whether a device is safe and effective for its use. So perhaps this is best explained with a couple of examples. And so what I have done here is I have chosen two examples to show, one, when the agency is looking back at safety and effectiveness, and two, when the agency is not and is relying more on what we call substantial equivalence, or in other words, we believe that if the device is equivalent to something that's already marketed, that something that is marketed is safe and effective, and therefore we're relying that by showing substantial equivalence, the device is safe and effective as the device that is being marketed. So in the two examples that I have chosen, first I've chosen a example of a device that we regulate under Class 2, 510K. That device, in this case, is a glucose meter. So I've shown you, I've taken the intended use of a glucose meter that we cleared. That is the language we use when we review under the 510K paradigm. So we take glucose meters for over-the-counter use. We clear them under the 510K paradigm. And this is the type of evidence that we would look to see whether a glucose meter is both analytically valid and, by extension, in many ways, clinically valid. Clearly, glucose meters or glucose as an analyte for the managing of diabetics has a lot of data supporting that managing of glucose values improves outcomes of diabetics. So the clinical validity of measuring glucose is not in question. It is well founded in the literature. So the agency does not typically ask a manufacturer to prove that by measuring glucose, the manufacturer can help a diabetic manage their diabetes and result in positive outcomes. So in this case, we have I'm just putting the intended use of a glucose meter, the One-Touch Ultra Plus, and what we can see here is that the intended use is dual. It's a little complex for these in that it is not only for the use of quantitative measure of glucose in fresh capillary and whole blood that determines essentially a sample type. The second part, the One-Touch Ultra Monitor, is not intended for use. It's intended to be used by a single patient and should not be shared. It's really more of a safety part of this. That is, we have seen that glucose meters that are shared they're not typically designed for to be cleaned in such a way that you guarantee that no blood-borne disease can be transmitted. So we usually make them in the intended use, state whether this is a meter that is to be used in multiple patients or not, what the design of the meter is. As it continues, the intended use tells you that it is intended for self-testing outside the body, that's in vitro diagnostic use, by people with diabetes at home as an aid to monitor the effectiveness of the diabetes control. That clearly is the intended use. This has further clarification, that is that the meter is not to be used in diagnosis or screening of diabetes or for neonatal use. The reason that those warnings are there is because even though there is plenty of data that glucose measurements can be used for such things as diagnosis or screening for diabetes or for neonatal use, the meter that is in hand has not been evaluated at the appropriate places to be able to determine whether it can safely be used for diagnosis or screening of diabetes or neonatal use. For diagnosis or screening, you need a very accurate and precise meters. Handheld glucose meters typically are not sensitive enough and will not give values that allow you to diagnose diabetes. Equally so, at least for this meter, the neonatal use requires the ability to measure glucose at levels in which this meter was neither designed nor likely to be accurate, and therefore a warning that this is not for neonatal use is put on. And so here you see a little bit the interplay between analytical validity and clinical validity, because it clearly, if the meter itself cannot go down to the levels that are necessary for neonatal use, then the clinical validity for that meter has not been determined, but is purely determined by the analytical validity that we look at. So what do we see in terms of performance and validation for these meters? So we look at, and I'm sorry, this slide is a little blurry. So we look at, for example, this is mostly considered analytical validity. We look to see that how well the meter measures glucose concentrations below 75 milligrams per deciliter. That's the hypoglycemic range for adult diabetics, essentially, adults, period. And being able to get an accurate measurement in the hypoglycemic area is very important. So we make the manufacturers look or have samples in the hypoglycemic area, and we look at to see how well those measurements fall within a range, either 5 milligrams per deciliter, 10 milligrams per deciliter, and 50 milligrams per deciliter. That gives the user an idea of how well the device performs in that section. And then typically for glucose concentrations above the hypoglycemic, there is a similar variance that is allowed. That is, it's allowed to be, we look to see how well it is within 5% of what the value should be, within 10%, 15%, and 20%. We find that particularly for over-the-counter glucose meters, describing the data in this way is helpful to people who use it. It gives you an idea of the variability of the test and the accuracy of the test. Finally, we look to see how well the meter performs against a standard reference, and we do that by looking at doing a correlation, looking at the slope and the intercept, and it gives you an idea of how accurate the meter is. So for glucose meters in this case, essentially, these are all things that one would consider under analytical validity. You have an analyte, you have a well-defined analyte, and you're looking to see what the accuracy, the bias, and the precision is. You're looking to see, and furthermore, we look to see what other things. So typically for glucose meters, we make sure that there is no dependence on, or that the values are still accurate at different altitudes. There is actually an oxygen dependent in some of these meters, and therefore you want to make sure that if they're going to be used at a place with a high altitude, that they actually still give you accurate results. We look to see what kind of interferences with as the hematocrit changes. That usually is also well known. And there are other substances that may affect the glucose meter, so we make the manufacturers evaluate them in that sense. So this is a well-controlled meter. We actually do a fair amount of reviews for these meters, and we make the manufacturers in a fairly robust manner test their devices to make sure that they work appropriately. Now, for a second example, I chose a case in which we're looking at a test that is either new or in class 3, and for which, again, in this case, we are going to be requiring clinical data, typically, in a form of a clinical trial or not, to determine the clinical validity of the test. And so here we had a test system that it was supposed to help determine whether a determine the assessment of moderate or severe acute kidney injury. And the reason to have this test is that you want to be able, if there's an acute kidney injury, you want to be able to determine whether the patient is going to progress to either it could progress to death or to CDK to a condition in which this becomes chronic kidney disease. And in either case, either death or chronic kidney disease, if you can identify the patients that are more likely to go towards that, you can be more aggressive with your treatment than if you have patients that are not likely to proceed to chronic kidney disease or to death. So having that test that will allow you to be able to have a prognosis in your patient is important and useful in this case. And this was a test that came in for just that assessment. So what kind of data do we see? So this is a real clinical trial, and there were actually 407 patients that were assessed with this score. And then it was determined the status later on as to whether there was acute kidney injury or no acute kidney injury, and the performance of the test was evaluated that way. As you can see, you look to see what we do this type of table in which we look at what the test result was versus what the outcome for the patient was, and do a table in which we know how many true results, how many true positives, false positives, false negatives, and true negatives. In this case, for this test, the number of false negative results was fairly low. That is, if the score is below 3, it is unlikely that the patient actually has acute kidney injury. It has a lot of this test has the number of true positives is low percentage-wise. In reality, most of the results are false positives. 46% of the results are false positives. That's a large number of false positives. But in the context of how this test is being used, the large number of false positives is not typically a problem. So when we look at this test, a test that identifies patients at risk for acute kidney injury, may aid in the prevention of progressive kidney disease or possible death, then what are the risks and benefits? A false positive result may result in a patient being misclassified into, this is the opposite. A false negative result may result in a patient being misclassified in a low risk, potentially resulting in temporary delay of implementation of kidney-sparing strategy. And a false positive result may expose a patient to aggressive treatment with toxic effects. And therefore, it is important to in this case, clearly, for patients that can progress to a deadly or complicated disease, one prefers to call the benefit-risk ratio is such that one would like to get false positives if possible, and not false negatives. So when we look at the risk of this test, we think that the test is safe and effective as given because people can actually make the right decisions. So just to wrap this up, therefore, the agency looks to see whether, depending on whether it is a class 2 test of which the risks are to the patient lower, and for which there may be already lots known about the analyte or the test and the clinical validity, then typically the agency mostly looks for studies that determine the appropriate analytical validity for the device based on what is known about the tie between that analyte and the clinical condition. For tests that are either high risk or that are new, and the determination as to whether there is a tie between the analyte that is being measured and the specific disease. For those tests, the agency actually does a review of the safety and effectiveness of the test and determines whether the test is appropriate, whether the benefits outweigh the risk, and therefore whether the test should be approved to go on the market. And that is essentially the basis of what the agency does when looking at in vitro diagnostic tests. And now, if there are any questions, I'll take a question. Thank you.
Thank you, Dr. Gutierrez. Thank you for bringing that important information to us. Before we get started with questions, I want to remind the audience how to reach us today. Questions can be sent via the green Q&A button at the lower left of the presentation window. We'll get to as many of your questions as we can today, and if we don't, we will follow up with you. Now, we do have our first question for Dr. Gutierrez. An attendee asks, does the FDA assess the clinical validity for all in vitro diagnostic tests?
So no, in truth, the FDA at this point is not assessing the clinical validity for all tests. The agency, when the law passed in 1976, took a look at the risk of the tests that were out there, and because of resources and other factors, decided not to regulate tests that were being developed by clinical laboratories. Clinical laboratories at that time typically developed tests that were used mostly in laboratories that were associated with hospitals and or academic centers, and they were being developed to meet some unmet need or to try to diagnose a rare or unusual condition. And the agency felt that those types of tests at that point were likely well controlled, and the FDA did not need to assess the clinical validity of such tests. The landscape has changed tremendously. There are now many thousands of what are called laboratory-developed tests. The agency does not typically assess the clinical validity for those tests. Laboratories in 1988 began to be regulated by the law that is called CLIA, which is administered by CMS. Under CLIA, laboratory inspectors that go into laboratories to inspect the laboratories, look to see whether the laboratory has validated the test. Typically, they look for analytical validity, but they do not typically review the clinical validity of that test. So many of the tests that are provided as laboratory-developed tests currently are not being regulated by the FDA and are not being regulated by anybody as to the extent of whether they're clinically valid or not.
Okay, thank you. We have another question. Is clinical validity evaluated similarly for other diagnostic tests?
So the agency regulates other tests that are not in vitro diagnostic tests. Scientifically, the way we regulate all those tests are similar. There are some differences depending a little bit on whether one can understand the analytical validity of the test through what kind of testing, whether it needs to be like it is with in vitro diagnostics with clinical samples or not. But in general, when we're looking to see whether our test is safe and effective, we are looking at very similar things. So, for example, when a new mammography device wants to come through the agency to be used for screening in mammography, then the assessment for that new device will be done in a very similar way to the way we do in vitro diagnostics.
Great. We do have another question. An attendee asks, I have heard the term companion diagnostic. Can you explain what that is?
So as we have learned more and more about specific biomarkers for specific diseases, and as the pharma has begun to be able to target the drugs to specific pathways that cause disease, we are now in an area in which, as you may have heard, it's either called precision medicine or personalized medicine, where there is a need for a diagnostic to select the population for that is going to benefit or not be harmed by a specific drug. So now the effectiveness of the drug is tied to a certain extent the effectiveness and the safety maybe also of a particular diagnostic that it needs to be used to determine who's going to take that drug. So if you need a diagnostic to tell you which population is going to benefit from a drug, we call that diagnostic a companion diagnostic. And we evaluate, again, it is a little bit similar in the way we go. First of all, we actually believe that in most cases those companion diagnostics are class 3, because it determines whether you're going to be taking a drug or the risk of taking that drug. And in most cases, drugs can be highly beneficial, but can also be highly problematic. So determining properly who's going to be taking that drug and the risks of that drug get translated to the device, if you like. And so when we approve drugs now that require the use of a particular diagnostic, we also approve the diagnostic at the same time. Now, because the only way to determine whether a drug is safe and effective typically is through randomized trials, then that evidence that is built to approve the drug, that is the randomized trials, is also what is used to determine whether the diagnostic is working properly, obviously, and therefore that evidence is used as the clinical validity of the diagnostic itself.
Great, thank you. Another question, is clinical validity for a companion diagnostic assessed similarly as for other in vitro diagnostic tests?
So I guess I took the question further. Then yes, as I just explained,
yes.
As we go, as we we do assess it similarly. The data sets tend to be much more. So we as with other diagnostics, we clearly look at the analytical validity of the test. Is the test measuring the appropriate analyte, and is it measuring repeatedly and accurately? but then we also use the data that is collected in the drug trials to determine whether the drug is effective as a sign as to whether the diagnostic was able to determine which populations were appropriately defined for taking the drug. We have had cases, interestingly enough, where the drug was considered to be probably likely beneficial for a particular population, and the clinical trials were done with the diagnostic to determine whether the drug was effective and whether the diagnostic actually could tell the difference, could select the appropriate population. And we have seen cases in which, as the data comes in, it turns out that the drug is effective on not just the target population, but in the other population. And in those cases, the diagnostic actually has then not become a companion diagnostic. It has not necessarily been approved by the agency because it becomes clear that the drug itself is useful for all comers and not just for part of the population.
I see. That is all the time that we have for questions today, but I would just like to ask Dr. Gutierrez if he has any final comments for our audience today.
I hope that I was able to get the point across that the agency determines whether a device is safe and effective or is substantially equivalent to other devices that are out there based on the analytical and clinical ability, and what the analytical and clinical ability is, or what we look for in the analytical and clinical ability, changes in many cases based on the knowledge that is out there and what the manufacturer needs to make the link between the accuracy of detecting the particular signal, the particular analyte, and the clinical use of that signal for either diagnosing or for managing or for any of the different uses that diagnostics are used.
Great, important information to bring to our audience today. I'd like to once again thank you, Dr. Gutierrez, as well as LabRoots for bringing this presentation today. Today's webcast will be available for on-demand viewing through October of 2017. Attendees today will receive an email from us alerting you when it's available on demand and posted on labroots.com. You're welcome to forward this announcement to any colleagues who weren't able to tune in live today. Thank you for logging on and participating in today's broadcast. We hope we see you next time. Goodbye.